science fiction, like a lunar lander or some, alien spacecraft. So again, we have the head, we have the tail. The head is an icosahedral capsid made up of protein protecting the phage DNA. This tail is incredible. These little tail fibers at the very tips are special proteins called adhesins. So it's the tail fibers that will attach to the surface of the host organism. Here is going to be E. coli. And it's very specific. The adhesins are going to bind to polysaccharides on the lipopolysaccharide of the outer membrane of the E. coli. And when the phage first attaches to the cell wall of the E. coli, the base of the tail will settle, and see these little spikes? It kind of anchors the base of the tail into the outer membrane, kind of holding the virus in place. Now, this is just the steps in replication, you guys, and what I'm going to do is I'm just going to fast forward to the actual cartoon. So we'll just walk through it. All right, so just to get you oriented, this is our E. coli. This is the cytoplasmic membrane of E. coli. This is the inside of our E. coli. This is the cell wall. Oops, and that's really bad, you guys, because that cartoon, it should only go up there. This is cytoplasmic membrane. This is the cell wall. Thin layer peptidoglycan, and our outer membrane, and the outer leaflet of the outer membrane has a lipopolysaccharide. These little purple things with tail, that's a lipopolysaccharide. So we can see that the phage is attaching to the cell wall of E. coli. We can see that the tail base plate with the spikes have settled in, and the spikes are punching into the outer membrane. And this is, wow, you guys, the phage releases a lysozyme-like enzyme. And the lysozyme-like enzyme is going to come down and cleave glycosidic bonds of peptidoglycan, weakening it. And then this next step is so wild, you guys. This tail shaft acts as a hypodermic syringe and needle. So we're going to get contraction of the tail, and that drives an inner protein shaft through the cell wall, through the cytoplasmic membrane, into the bacterium. I mean, this thing is acellular. How sophisticated is this? Isn't that the craziest thing? So here's this inner shaft acting like a needle, a hollow needle, through which then the phage DNA is passed across the cell wall, across the cytoplasmic membrane, and into the cytoplasm of the bacterium. Now, this bacterium is a goner, right? You guys remember that original description of viruses is trouble wrapped in protein? This is trouble. This is death for the little E. coli. Shakespearean tragedy. Okay, so now we'll go to your cartoon, and we'll walk through some of the basic steps. And again, folks, depending on the textbook, they might describe the steps. They might give them bold titles a little bit differently. But the same basic steps are going to occur in the lytic replication cycle. Lytic because the bacterium is going to end up dying from lysis. So here, the first step is attachment. Okay, a fancy term for attachment of a virus is adsorption. Why they had to come up with a different name for attachment, I don't know. But when viruses attach, virologists say we have adsorption of the virus to the host cell. Here's our little E. coli victim, and this is the E. coli chromosome. So we're just kind of looking at main events of the host cell. So our phage attaches or adsorbs. We then have injection of the phage DNA, which in this author they're calling it entry. Okay? And what happens is when the phage DNA enters the E. coli, the E. coli RNA polymerase transcribes the phage DNA into phage mRNA, and then the E. coli ribosomes translate the phage mRNA into enzymes and phage proteins. Some of those phage proteins are enzymes that will hydrolyze, break the bacterial chromosome into pieces. And the reason is the bacterial chromosome nucleotides will be used as building blocks to make copies of the phage DNA. This is the perfect parasite, you guys. Meanwhile, the E. coli is no longer taking care of itself. It has become, as Dr. Meyer says, a phage-producing factory. That's all it's doing now, is making phage DNA and phage proteins. It's no longer taking care of itself. So here we see biosynthesis, meaning that the poor little bacterium, all it's doing now is making copies of phage DNA. making phage proteins. The wild thing is, you guys, the phage proteins self-assemble. It's like the Twilight Zone. It's the craziest thing. The phage proteins self-assemble into capsids and tail components, and if that's not wild, then the phage capsids will start packaging pieces of DNA from the cytoplasm. Now, at this point, most of the pieces of DNA in the cytoplasm will be phage DNA, but you guys, can you imagine that occasionally, accidentally, a piece of bacterial host DNA might get packaged? Does that make sense to you? Yeah. Remember that generalized transduction we were describing? Yeah, this is where we could get generalized transduction occurring. Following packaging of the DNA into the phage heads, again, crazy, you guys, the tails self-assemble, they organize and attach to the capsid, and so now we have fully assembled phage, and at this point, the phage release that lysozyme-like enzyme that will, again, weaken the peptidoglycan. And the final step here is lysis of the little host E. coli and release of all these infectious phages. So hence, lytic cycle, right? So lytic cycle, you guys, the E. coli always dies, and we get replication of newly infectious phage, right? Lytic cycle. So T even phage, lytic cycle only. Yeah.
This one's a little different. So should we get to know that one more, or this one?
If you can, yeah, I understand. Right, what Dylan's saying, folks, because they've got down here, they've got absorption and penetration as step 1 and 2. So see, we can see how different language is being used. So for attachment, they're using adsorption. For entry, they're using penetration. And let's go over here. So biosynthesis is the same. And instead of assembly, they're using maturation. So you know what I would suggest, you guys, is it's really good, because when you go out into your careers, you're going to be working with folks that have had different teachers, different courses. The more flexible you are, it's like if they're using the term maturation, talking about viruses, you know, oh, maturation, that's assembly. The more efficient you're going to be. So you're not going to like this. What I recommend is on this handout, you put absorption equals attachment. Penetration equals entry. Biosynthesis is the same. Maturation is assembly. Does that make sense? I know it doesn't make you happy, but I think it's going to make you more efficient out there in the real world. And usually on a lecture exam, usually I try to write the questions so it won't be too confusing. It'll be kind of obvious which one it should be. Yeah. I am sympathetic, you guys. I know you have so much vocabulary to remember, right? Okay, so just the steps in assembly. And what we'll do is we'll just introduce you guys to the second type of replication cycle. Lytic phage can't carry out this replication cycle, which is called the lysogenic cycle. So just really quickly, we're going to introduce a second group of bacteriophage called temperate phage. And the most famous temperate phage is the temperate phage of E. coli called lambda. So this is lambda phage. And the beauty of lambda phage is they have a choice. They can either carry out either the lytic cycle or a brand new one, the lysogenic cycle. And just really quickly, guys, let me see if I can just give you an idea of this. Okay, so in the lysogenic cycle, the first two steps are the same. We have attachment or absorption. We have entry or penetration. You'll notice, you guys, on the lambda phage, it's got a capsid, the tail shaft. It has short little tail fibers. It's just not as pretty as the T-even phages. So here's the phage DNA entering. Now here's the important step, you guys. Step 3 is where things change. In the lysogenic cycle, instead of the phage DNA being transcribed and translated to make phage proteins that are going to destroy the bacterial chromosome, instead, the phage DNA inserts itself into the bacterial chromosome. Important vocabulary word. Phage DNA inserted into the bacterial chromosome is called a prophage. Now, to the E. coli DNA polymerase that's going to copy that chromosome, that prophage is part of the chromosome. So when the E. coli is getting ready to divide in two and the chromosome is copied, will the prophage DNA be copied as part of the chromosome? Yes. So that means that generation after generation, that prophage will be passed on to future generations as part of the E. coli chromosome. Right? Now, what we'll discover on next Tuesday is when there's damage to the DNA of the E. coli, the phage can cut itself out of the chromosome and then trigger the lytic cycle. This is called induction, right? So this, you guys, is where we'll start on Tuesday. We'll start with an introduction to this incredible process of induction, where the phage can switch from lysogenic to lytic cycle. Okay, guys.